Okay students, today you are going to study next cell organelle, that is ribosome. After this we will study one more cell organelle, that is nucleus. So today you are going to study first cell organelle, that is ribosome. As I said earlier, this is called as a protein factory because this is mainly involved in the synthesis of protein. As far as ribosome is concerned, two types of ribosomes are found in two different types of cells, like the eukaryotic cell and the prokaryotic cell. So you can see the comparative study of the ribosome present in the prokaryotic cell and the eukaryotic cell. First see the ribosome of the prokaryotic cell, then we will study a ribosome of eukaryotic cell. So each ribosome consists of two parts. First part is called as a small unit that we can see here. Then the second part is called as a large unit. Okay. So both types of cells show similar type of structure of the ribosome that we can see. This is a typical ribosome. But eukaryotic cell contains 80S type of ribosome, while prokaryotic cell contains 70S type of ribosome. Let us see the small unit of the prokaryotic ribosome or the ribosome present in the prokaryotic cell. Then we will see a large unit of ribosome present in the prokaryotic cell. Then we will study the ribosome present in the eukaryotic cell. Let us see a small unit of prokaryotic cell. Okay, small unit of ribosome present in the prokaryotic cell. So this small unit is basically made up of 16S rRNA plus 21 proteins. So this small unit is basically made up of two things, rRNA and the proteins. Okay, so 16S rRNA plus 21 proteins. Okay, 21 proteins. Number of proteins are 21. So this is basically a small unit of ribosome present in the prokaryotic cell. Let us move towards the large unit of ribosome present in the prokaryotic cell. So this contains, large unit is made up of 23S R small r, that is ribosomal RNA, then 5S ribosomal RNA plus 34 proteins. Proteins are now more and the number is 34 here. This small unit contains only 21 proteins, while this large unit contains 34 proteins, 5S rRNA and 23S rRNA. Okay, अशा पद्धतीने हे तिघांच्या मिळून हा काय होतो, एक लार्ज युनिट तयार होतं. ओके, अशा पद्धतीने हे स्मॉल युनिट आणि हे स्मॉल लार्ज युनिट हे दोन्ही एकत्र आल्यानंतर जो तयार होतो तो रायबोझोम, की जो कोणामध्ये पाहायला मिळतो, प्रोकॅरियोटिक सेल मध्ये. सो ribosome present in prokaryotic cell is made up of a small unit plus a large unit. Small unit is basically made up of 16S rRNA plus 21 proteins. Large unit of this ribosome is made up of 23S rRNA plus 5S rRNA plus 34 proteins. Let us move towards the, and after this combination, this becomes a 70S type of ribosome. So this is a typical structure of a complete ribosome. Let us move towards the next one, that is the ribosome present in the eukaryotic cells. So basically a small unit of this type of ribosome is made up of 18S rRNA, 33 proteins. Okay, this becomes a 40S unit, while this one becomes a 30S units. Okay, in as far as large unit of this ribosome found in the eukaryotic cell is basically made up of 28S rRNA plus 5.8S rRNA, that is ribosomal RNA, plus 50 proteins, and this total becomes a 60S type of 60S. So this 40S plus 60S, this becomes now a 80S type of ribosome. Understood? So again, this is a typical ribosome found in the eukaryotic cell. Okay, so this is simply regarding the structure of ribosome. As I said earlier, this ribosome is called as a protein factory because this ribosome is mainly involved in the synthesis of protein. That's why this is called as a protein factory. This ribosome was first time observed by scientist Palade in 1953 under electron microscope. Okay, this ribosome lacks a membranous cover. There is no protective cover around a extra cover around this ribosome. So this ribosome lacks a membranous protective covering. As far as the eukaryotic type of ribosome is concerned, in eukaryotic cell it is present in the mitochondria, cytoplasm, plastids, etc. And it is, as I said earlier, eukaryotic cell मध्ये कोणत्या प्रकारचा रायबोसोम असतो 80S, तर प्रोकॅरियोटिक सेल मध्ये कोणत्या प्रकारचा रायबोसोम असतो तर 70S. Okay, then this ribosome is mainly connected to the ER, that is endoplasmic reticulum. So bound ribosome, that type of ribosome which is connected to the ER, that is endoplasmic reticulum, is called as a bound ribosome. So this bound ribosome is mainly concerned with the protein synthesis. Once a protein is synthesized, then it is transported towards the cytoplasm outside a cell, not a cytoplasm, outside a cell. Okay, after processing by the endoplasmic reticulum and the Golgi complex. Let us move towards a specific or a different type of cell, that is the acinar cell of the pancreatic gland produces a digestive enzyme. I repeat, a specific or a special type of cell, that is the acinar, ACINAR, acinar cell of pancreatic gland produces a digestive enzyme which breaks down a complex substance. Okay, and this is simply regarding the type of protein. So that type of protein which helps to break down a complex material into the simple one is called as an enzyme. So as I said earlier, all enzymes are protein, but all proteins are not enzyme. I repeat my sentence, all enzymes are proteins, but all proteins are not enzyme. So free ribosomes, it means that those who are not attached to the ER, that is endoplasmic reticulum, are known as free ribosomes. So free ribosomes come together and they produce a chain-like structure which is called as a polyribosome. Okay, so they produce a polystructure. Then the most important thing is that the free ribosomes as well as bound ribosomes can change their role as well as their functions, okay, according a situation. So this is simply, and they can also change their positions also and the functions. So this is simply regarding a structure of a ribosome. Okay, 70S type of ribosome found in the prokaryotic cell and the 80S type of ribosome found in the eukaryotic cell. This cell organelle was first time discovered by scientist Palade in 1953 under electron microscope. So this is a typical structure of a ribosome found in prokaryotic cell as well as in eukaryotic cell. And this ribosome is called as a protein factory because this is mainly involved in the synthesis of protein. Now just move towards the next and the most important cell organelle, that is a nucleus. As I said earlier, this nucleus is called as a master cell organelle because this nucleus is mainly involved in the synthesis of DNA, in the synthesis of protein, in the synthesis of a ribosome. That's why this is called as a master cell organelle of the cell. This becomes a distinct and very clear in a interphase. Okay, so this is the structure of nucleus taken from the interphase. So this is called as a master cell organelle. Let us see a structure of this nucleus. Now a typical nucleus consists of a outer and the inner membrane. Okay, now this is a outer membrane, this is a inner membrane. So this typical nucleus consists of membrane or the nuclear membrane. This consists of inner membrane as well as outer membrane. Then the dark stained body is called as a nucleolus. Then this is called as a chromatin network. Later on, after condensation, this reveals into a definite number of chromosomes. Okay, so this is a network of chromatin. This is called as a chromatin network also. Okay, or the network of chromatin. Then this is called as a karyosome. Okay, karyosome. Okay. Then the most important thing is that this remaining part or the viscous matrix is called as a karyolymph. Okay, karyolymph. Okay, त्याला म्हणतात karyolymph असं म्हणतात. Okay, let us move towards the few more points regarding this nucleus. So these two membranes are separated from each other by 5 to 50 nm, that is nanometer. Now this membrane is not continuous. This membrane, outer membrane as well as inner membrane, is not continuous. This outer and the inner membrane or the nuclear envelope or the nuclear membrane is interrupted by a small opening which is called as a nucleopore असं म्हणतात. त्याला काय म्हणतात? Nucleopore or the nuclear pore. This is called as a nuclear pore. This you can see a nuclear pore here nuclear pore. You can notice this is also called as a nuclear pore. So this is not a continuous membrane. This is interrupted nuclear envelope, and this is interrupted by small openings which are called as a nuclear pores. Okay, let us see the outer membrane and the inner membrane. Then this is called as a nucleoplasm or a karyolymph. Okay, let us move towards the few more points of this particular cell nucleus, that is a main or the master cell organelle. Okay, this nucleoplasm contains, most important thing is that this nucleoplasm contains salts, minerals, nucleic acids, proteins, ribosomes, as well as nucleolus and the minerals. Okay, न्यूक्लिओप्लाझम मध्ये काय काय बघायला मिळतं? सॉल्ट्स आहेत, मिनरल्स आहेत, न्यूक्लिक ऍसिड आहे, त्याचप्रमाणे त्याच्यामध्ये न्यूक्लिओलस आहे, प्रोटीन्स आहेत. अशा पद्धतीने याच्यामध्ये वेगवेगळे प्रकारचे घटक बघायला मिळतात. ओके, दिस न्यूक्लिओप्लाझम और ए कॅरियोलिम्फ contains a network. You can notice this one. This is a network of a chromatin or chromatin network. Now this chromatin network undergoes a process that is called as a condensation process. Okay, after condensation this network appears like this. So some part of this appears dark while some part of this appears a thin portion. Okay, so those parts which are appear thick are known as a euchromatin. Dark parts are known as heterochromatin, while thin parts are known as euchromatin. So this heterochromatin is inactive. This contains large amount of proteins and less amount of DNA. This euchromatin is a active part of the chromatin network. This contains large amount of DNA and less amount of proteins. Okay, and vice versa. बघा, हा इनएक्टिव्ह आहे, याच्यामध्ये लेस अमाउंट डीएनए आहे, मोठ्या प्रमाणामध्ये प्रोटीन्स आहे. So this is heterochromatin. A dark stain or the more condensed chromatin network is called as a heterochromatin, which is basically inactive due to the presence of less amount of DNA and large amount of protein. This is euchromatin. This euchromatin is a thin chromatin network which contains large amount of DNA and less amount of protein. Okay, that's why this becomes a active portion of the chromatin network. As I said earlier, this undergoes a condensation process and this reveals into definite number of chromosomes. As I said earlier, after condensation each chromosome appears a bivalent, consist of two chromatids. Okay, and these two chromatids are held together by a centromere. Understood? Now just move towards the most important one more thing, that is the nucleolus. So this nucleolus हे बघा न्यूक्लियस मध्ये हा पूर्ण न्यूक्लियस आहे की त्याला मास्टर सेल ऑर्गनल म्हणतात. या न्यूक्लिओलस मध्ये अशा पद्धतीने डार्क स्टेन बॉडी बघायला मिळते, याला म्हणतात न्यूक्लिओलस. बघा याच्यामध्ये हा पूर्ण झाला न्यूक्लियस. आय रिपीट माय सेंटेन्स, दिस एंटायर बॉडी or cell organelle is called as nucleus. Understood? This is called as a master cell organelle because this is mainly involved in the synthesis of DNA, RNA and the protein. That's why this is called as a master cell organelle. This consists of a whole genetic information which leads to the variation and inheritance. अंडरस्टूड? त्याच्यामध्ये होल जेनेटिक इन्फॉर्मेशन आहे की कशासाठी आवश्यकता आहे त्याची, किंवा त्याचा काय रोल आहे या होल जेनेटिक इन्फॉर्मेशनचा. सो दिस होल जेनेटिक इन्फॉर्मेशन प्लेज अन इम्पॉर्टंट रोल इन दी हेरिडिटी अँड दी व्हेरिएशन ऑफ each and every species. Understood? As far as human body is concerned, our each and every cell contains how many sets of chromosomes? 23 sets of chromosomes. Means total number of chromosomes are 46. Out of 46 chromosomes, there are 22 pairs of autosomes and 1 pair of sex chromosomes. Whether it is XX or XY, that depends on whether it is male or female. Okay. So this is simply regarding a master cell organelle, that is a nucleus. Okay. As I said earlier, this is called as a nucleus. Now this nucleus contains a nucleoplasm, a viscous fluid. This contains also a nucleic acid, that is a chromatin network in the network-like structure. This shows dark bodies as well as thin structures. Dark structures are known as heterochromatin, while thin structures are known as euchromatin. After some period, this entire genetic information or this chromatin network undergoes condensation process and it reveals into definite number of chromosomes. Understood? That depends again on the species. Human सेलचा जर हा एक न्यूक्लियस असेल तर याच्यामध्ये 46 क्रोमोझोम्स बघायला मिळतील कंडेन्सेशन झाल्यानंतर. ओके. हा आता याच्यामध्ये आणखी एक महत्वाचा जो न्यूक्लिओझोम मध्ये किंवा कॅरिओलिम्फ मध्ये जो सेल ऑर्गनल बघायला मिळतो, याच्यामध्ये न्यूक्लियस मध्ये जी डार्क स्टेन बॉडी बघायला मिळते त्याला म्हणतात न्यूक्लिओलस. त्याला म्हणतात न्यूक्लिओलस. हा न्यूक्लिओलस न्यूक्लियस मध्ये असतो, न्यूक्लिओलस. नाउ दिस न्यूक्लिओलस इज मेनली इन्वॉल्व्ड इन द सिंथेसिस ऑफ RNA as well as protein, and it is basically made up of itself. It is made up of RNA and the protein, and this also involved in the process of synthesis of RNA and protein synthesis. So this dark stained body is called as a nucleolus. Okay. Then you can see a karyosome also in this particular nucleus. Okay. So this nucleus is very large in oocytes and it is very small in the sperm. सगळ्यात छोटा जो न्यूक्लियस आहे तो याच्यामध्ये स्पर्म सेल्स मध्ये, आणि सगळ्यात मोठा जो न्यूक्लियस आहे तो oocyte मध्ये बघायला मिळतो. ओके. तर अशा पद्धतीने आपण आज हे न्यूक्लियसचं स्ट्रक्चर बघितलं आणि त्याचप्रमाणे आपण रायबोझोमचं स्ट्रक्चर सुद्धा पाहिलं. परत एकदा बघूया, आपण ओव्हरलुक करूया. हा जो रायबोझोम आहे याला प्रोटीन फॅक्टरी असं म्हणतात. ओके. कारण रायबोझोम हा मेनली प्रोटीन सिंथेसिस मध्ये इन्व्हॉल्व्ड असतो आणि म्हणून याला काय म्हणतात, प्रोटीन फॅक्टरी असं म्हणतात. की पॅलेटने ही पहिल्यांदा डिस्क्राईब केला इलेक्ट्रॉन मायक्रोस्कोपच्या माध्यमातून. त्याने पॅलेटनी 1953 मध्ये हा डिस्क्राईब केला. त्याला म्हणतात रायबोझोम. आता बघा, प्रोकॅरियोटिक सेलमध्ये पण रायबोझोम बघायला मिळतात, युकॅरियोटिक मध्ये पण पाहायला मिळतात. प्रोकॅरियोटिक मधला जो रायबोझोम आहे त्याच्यामध्ये आता टिपिकल जो रायबोझोम आहे तो दोनच युनिटनी बनलेला असतो. टिपिकल रायबोझोम इज बेसिकली मेड अप ऑफ 2 युनिट्स. upper unit is called as a smaller unit or the small unit, and the lower one is called as a large unit. पण याच्यामध्ये नंबर ऑफ प्रोटीन्स आणि युकॅरियोटिक मध्ये नंबर ऑफ प्रोटीन्स, त्याचप्रमाणे आर एन ए याच्यामध्ये डिफरन्स बघायला मिळतो, आणि म्हणून अशा पद्धतीने जो बनतो त्याला म्हणतात रायबोझोम. बघा, प्रोकॅरियोटिक मधला जो रायबोझोम आहे, म्हणजे प्रोकॅरियोटिक सेल मध्ये असलेला जो रायबोझोम आहे, त्याच्यामध्ये जे स्मॉल युनिट आहे, स्मॉल युनिट 16S RNA प्लस 21 प्रोटीन्सनी बनलेला आहे. याच्यामधला जो लार्ज युनिट आहे तो 23S RNA आणि 5S RNA प्लस 34 प्रोटीन्सनी बनलेला आहे. अशा पद्धतीने हे 30 युनिट वरचं आणि हा 50 युनिट एकत्र आल्यानंतर 80 होतात, 80S होतात, पण 10 मायनस करून तो जो राहतो शिल्लक त्याला म्हणतात 70S रायबोझोम. So 70S type of ribosome is mainly found in the prokaryotic cell. It is only present in the 70S type of ribosome is only present in the prokaryotic cell. Now just move towards the eukaryotic type of ribosome. याचा जो 40S युनिट आहे, पहिला जो युनिट आहे तो 40S स्मॉल युनिटला 40S युनिट म्हणतात, की जो 18S रायबोझोम्सनी बनलेला आहे आणि 33 प्रोटीन्सनी बनलेला आहे स्मॉल युनिट, त्याला म्हणतात 40S युनिट. ओके. लार्ज युनिट जो आहे तो 60S नी बनलेला आहे, 60S, आणि हा 60S कशाने बनलेला आहे, 28S RNA rRNA, 5.8S rRNA परत, आणि 50 प्रोटीन्स. याच्यामध्ये बघा 50 प्रोटीन्स आहेत, तर याच्यामध्ये किती आहेत, 34 आहेत. सो दिस बिकम्स ए 60S टाइप ऑफ लार्ज युनिट. 60 प्लस 40 असे एकत्र येऊन याच्यामध्ये किती होतात, ते 100 होतात, पण ऍक्च्युअली 100 नाही आहे, हे किती होतात, 80S. सो 80S टाइप ऑफ रायबोसोम इज प्रेझेंट इन, इट इज ओनली प्रेझेंट इन, इट इज रेस्ट्रिक्टेड इन युकॅरियोटिक सेल. तर अशा पद्धतीने हा आपण रायबोझोम बघितला, हा कम्प्लीट रायबोझोमचं स्ट्रक्चर अशा पद्धतीने बघायला मिळतं. ओके. त्यानंतर आपण मास्टर मॉलिक्यूल बघितला. व्हाय दिस इज कॉल्ड एज अ मास्टर मॉलिक्यूल? Because this nucleus is mainly involved in the synthesis of RNA, mainly DNA, and the protein also. That's why this is called as a master cell organelle. This encloses a whole genetic information in the form of chromosomes. I repeat my sentence. This nucleus encloses a whole genetic information of a organism. That's why this is called as, which plays an important role in the heredity and the variation. That's why this is called as a master cell organelle. This consists of a outer membrane that is called as a nuclear envelope or the nuclear membrane, outer membrane and the inner membrane. So the space present between the outer membrane and the inner membrane is 10 to 50 nanometer. Then this membrane is not a continuous membrane. This is interrupted by small openings, or the apertures are called as nuclear pore. Okay. This nuclear envelope encloses a network-like structure that is called as a chromatin network, as well as nucleolus, karyosome, and the viscous fluid is called as a nucleoplasm or the karyolymph. Okay. Then this network is basically contains, this entire network contains a whole genetic information for the synthesis of chromosomes. Okay. So that's why this entire network plays an important role in the heredity and the variation. This cell organelle is mainly involved in the synthesis of DNA mainly, as well as RNA, and the synthesis of protein. That's why this is called as a master cell organelle. This network shows some part of this network is dark, while some part of this network is less dark or the thin, and the some part of this network is thick. So thick part is called as a heterochromatin, which is less active, because this is basically made up of more proteins and the less DNA, while this euchromatin is a thin part of this network, that is chromatin network, which is mainly active part of this chromatin network, which is basically made up of large amount of DNA and small amount of proteins. So this is simply regarding a structure of a nucleus and a structure of ribosome. As I said earlier, this is bigger in size in case of oocyte and small in size in case of sperm. This is very small in sperm and very large in the oocyte. Okay, so this is simply regarding. So I hope all of you have understood all both these cell organelles, that is ribosome and the nucleus. ओके, आता याच्यामध्ये महत्वाचा जो पार्ट येतो तो येतो तर विद्यार्थी गफलत बघा कुठे करतात ते. न्यूक्लियस, न्यूक्लियसची स्पेलिंग वेगळी आहे आणि न्यूक्लियसची स्पेलिंग बघा NUCLEUS न्यूक्लियस आणि न्यूक्लिओलस NUCLEOUS न्यूक्लिओलस. बघा हा जो न्यूक्लियस आहे हा पूर्ण हा झाला सेल ऑर्गनेल झाला, त्याला मास्टर सेल ऑर्गनेल म्हणतात, बरोबर नाही. तर हा जो न्यूक्लिओलस आहे हा न्यूक्लिओलस या न्यूक्लियसचा एक पार्ट आहे की जो न्यूक्लिओप्लाझम मध्ये बघायला मिळतो. डार्क स्टेन बॉडी आहे की जो प्रोटीन आणि RNA बनलेला आहे. ओके, हा डार्क स्टेन बॉडी जो पार्ट आहे तो न्यूक्लियसच्या आतमध्ये बघायला मिळतो आणि हा न्यूक्लियसचा एक पार्ट आहे. समजते का? तर न्यूक्लियस हा पूर्ण एक बॉडी झाली, पूर्ण बॉडी झाली. ठीक आहे, याला मास्टर सेल ऑर्गनेल असं म्हणतात. तर याच्यामध्ये आणि न्यूक्लियस मध्ये गफलत करू नका किंवा कन्फ्युजन करून घेऊ नका. न्यूक्लिओलस हा न्यूक्लियसचा एक पार्ट झाला आणि न्यूक्लियस हा काय झाला, एंटायर सेल ऑर्गनेल झाला. ओके, सो आय होप ऑल ऑफ यू हॅव अंडरस्टूड ऑल दीज पॉइंट्स विदाउट ए डाउट. थँक यू. Next part, that is the centrioles and the remaining part of this chapter we will see in next lecture. Thank you.